Let's talk about meiosis, its sources of variation, and applications of meiosis. Remember that mitosis produces daughter cells that are genetically identical. Meiosis produces daughter cells with different combinations of genes. This variation is accomplished by gamete success. Only one sperm can fertilize the egg. Crossing over. It depends on the distance between genes and the length of the chromosome. Crossing over happens during prophase 1. So remember that the two sides of a duplicated chromosome are referred to as sister chromatids. Non-sister chromatids cross over and exchange genetic material. Collectively, the two homologous chromosomes are referred to as a tetrad. And the result is that we get 4 different chromosomes heading to each gamete. So explaining it in detail, crossing over occurs at random between pairs of homologous chromosomes. During prophase 1, homologous chromosomes form pairs or tetrads. Non-sister chromatids cross over each other and exchange segments of chromosomes. Chromosomes in the gametes contain new combinations of genetic material. Crossing over occurs more often between genes that are further apart. So let's look at, first of all, here's a chromosome, and the genes on it are A, B, and C. We refer to these 3 genes as linked genes because they are on the same chromosome. The only way they would not be transferred to the same gamete is if they crossed over. And the chance of them crossing over is greater the further they are apart because there's more distance between them. So A and C are more likely to cross over than between B and C because there's a very small distance between B and C in which to cross over. Independent assortment is the third source of variation for meiosis. It is the way one homologous pair lines up in metaphase 1, has nothing to do with the way that another homologous pair lines up. It is random. Okay, so independent assortment, the chromosome arrangement in metaphase 1 can be different every time. So just an example of 3 different ones, it could be that on one side we have 1 blue chromosome and 2 red. So we're going to assume that the blue is from dad, the red is from mom. So one set of gametes would get 1 blue from dad and 2 red from mom, and the other set would get 1 red and 2 blue. Or it might be blue, blue, blue in one, red, red, red in the other, or 2 blues and a red, and the other one has 2 reds and a blue. So again, every time the chromosomes align along the metaphase plate, the way they align individual pairs, the tetrads, is independent of the next tetrad. And that provides variation. Okay, let's talk about applications of meiosis. We're looking at a life cycle here of an animal. And if we start with meiosis, we know that the result of meiosis is haploid cells, or 1N. So we know that gametes are 1N. Fertilization, we know, doubles that because we are adding N plus N. So the zygote would be 2N. Mitosis keeps the numbers the same. So if we start out with 2N, we are going to end up with more 2N in the animal body. And that's how we label a life cycle. Let's take a look at a plant life cycle that has alternation of generations, which means it spends some of its time as a haploid individual. Let's start with meiosis again, because we know that it produces cells that are 1N. Mitosis keeps the numbers the same. So if we start out with 1N, we're going to end up with more cells that are 1N. Gametes still 1N. Here we have fertilization, so we know the zygote is 2N. Then through mitosis, if we start with 2N, we're going to end up with 2N. Next, let's look for, here's meiosis. So meiosis ends with 1N. Let's continue from there. Mitosis, we're going to have more 1N. They're the same. More mitosis, which means gametes are each 1N. Then we have fertilization, so the zygote is 2N. After the zygote, we have mitosis. That means the sporophyte is the same, it's 2N. And then more mitosis, meaning the plant is again 2N. Okay, so we're looking at the words we know: meiosis, fertilization, and mitosis. Okay, let's look at another plant life cycle. This time we're filling in just the 2 blanks. Here we have on the top, look at it's 1N, 1N, and then down here it changes to 2N. So 2N to 1N is meiosis. It produces 1N. And this other one must be fertilization, going from 1N to 2N. Let's take a look at the next one. Okay, so we have this clue. The top half of the life cycle is all 2N. The bottom half is all 1N. So going from gametes to a zygote from 1N to 2N, that's fertilization. And going from 2N to 1N, that would be meiosis. Before we get there, however, going from a zygote to a sporangium, that's growth. So that's, oh, we did do meiosis first. That's mitosis. Anywhere within the half of the life cycle, there is growth. So it's going to be mitosis in between. Okay, let's try another practice. Process A, going from 2N to 1N, is meiosis. Good. Process B, from 1N to 1N, it's the same. So that's mitosis. Process C, from 1N to another N, mitosis. And the last one, N to 2N, it's doubling. So that is fertilization. Okay, let's try another one. Let's start with meiosis. Produces spores, which are 1N or haploid. Mitosis keeps it the same. So the gametophyte is also going to be haploid. Now we have mitosis, so the gametes are haploid. And fertilization means the sporophyte is diploid. Okay, next one, we have the life cycle of a jellyfish. Where should we start here? Let's start with the zygote. We know that zygotes are 2N. And it grows into a larva, so that's also, it's mitosis, producing a larva, which is 2N. Metamorphosis is just rearrangement of the cells. It's forming into something else, but it's not making eggs and it's not fertilization. So that's more mitosis, which means the polyps are 2N. Then it says it's asexual reproduction. Asexual reproduction, remember, is an application of mitosis. So that is still 2N. We have more budding. Asexual reproduction is mitosis, producing 2N individual. That's growing, so that's mitosis to produce 2N. And here we have formation of eggs and sperm. That's meiosis. Eggs and sperm are 1N. And then forming a zygote is fertilization.